Hello, my name is Ian Kerman, and today I'm going to be presenting research that we have done on predicting antibody developability using machine learning. An antibody's developability index is a measure of the antibody's tendency to aggregate. Developability index, or DI, is calculated from the antibody structure and can then be used to rank different antibodies against each other. Determining the structure experimentally or computationally, however, can be expensive. Here we propose a method to predict the developability index of an antibody directly from sequence using machine learning. The overall workflow can be split into 4 distinct parts: featurization of the antibody sequences, labeling of the antibodies as having high or low developability, training the machine learning models, and lastly, evaluating the model's performance and finding the best performing model and hyperparameters. The workflow was implemented in BioVia Pipeline Pilot, a visual programming and pipelining tool that allowed us to automate the labeling of antibodies, featurize our sequences, as well as build and evaluate hundreds of different models. We use antibody structures from the Structural Antibody Database, or SABDAB. BioVia Discovery Studio, automated through Pipeline Pilot, was used to clean up both the PDB structure files and the sequences, and to calculate the developability indices of the structures. We classified 20% of the developability indices as highly developable, and the remaining 80% as low. Our machine learning model will then predict whether new antibody sequences would have a high or low developability. For featurization, we considered many different types of features for the antibody sequences, including physiochemical features calculated by emboss PepStats and XBASI-ProParam, and we also used learned embeddings implemented in Python. To efficiently search for the best set of features, models, and hyperparameters for predicting DI, we used Pipeline Pilot to combinatorially train models. We then used tenfold cross-validation to determine the best set of hyperparameters for each of the model types. And finally, after all of the models were built, we evaluated each model using a test data set and calculated confusion matrices and the area under the ROC curve for each of them. In total, 532 different models were built and evaluated, covering 6 different model types, plus a random control, and 76 different feature sets. In this heat map, we visualize the area under the ROC curve of each model, with the model type being given along the x-axis and the feature set along the y-axis. From this chart, you can see that both the physiochemical features and the embedding features with k-mer lengths of 2, 3, or 4 perform the best. We can also see that the support vector machine, random forest, and XGBoost model types outperform the multilayer perceptron, Gaussian Bayes, and logistic regression models. Overall, the best performing model was the random forest model with multiple sequence aligned physiochemical features. When we were evaluating our model's performance on the validation data and comparing that to its performance on the training data, we found indications that overfitting could be occurring. There is a steep drop in the sensitivity value in Cohen's kappa statistic when looking at the validation versus training data for different model types. Additionally, there is also an increase in the balanced error rate. We see a similar trend when looking at the random forest model across all of the different feature sets, and this is an area for improvement and something that we'll explore in future research. In summary, we've shown the feasibility of using machine learning to predict antibody developability from sequence. For more details, I encourage you to check out our preprint paper of the same title on bioRxiv. And lastly, I'd like to thank the students and professors at Harvey Mudd College who performed much of the work that you've seen today. Thank you and have a great day.